There's a certain comfort in the physical removal of a medical problem. Like you have a splinter, you pull it out, and the pain stops. You have a tumor, a surgeon cuts it out, and the immediate thought is just, well, it's gone.
Yeah, that is always the ideal scenario, right? I mean, surgical resection has been the gold standard for solid tumors for decades, but the reality of oncology is just far less clean than removing a splinter.
Right, and that brings us to the core of today's deep dive. We are looking at this fascinating 2026 study from Cell Reports Medicine by Zhang and colleagues, and it really focuses on what happens in that surgical cavity after the tumor is physically removed. Because, tumors don't have these smooth, clean edges.
No, not at all. They have these highly irregular, very invasive margins, which means, practically speaking, microscopic residual cancer cells are almost always left behind in the tissue.
Yeah.
And the environment they are left in is fundamentally altered by the surgery itself.
And this is the part that genuinely shocked me when I was reading the research. The very act of cutting the tissue, the trauma of the surgery that's meant to cure you, it actually creates a localized environment that helps those leftover cancer cells thrive.
It's a massive biological paradox, honestly.
It really is. It helps them hide from your immune system.
Exactly. The trauma provides the exact fuel the remaining cancer cells need to mount this devastating comeback.
So by the end of this deep dive, you're going to understand how scientists are finally fighting back against this. They've developed a laser-activated shape-shifting hydrogel that literally melts into surgical wounds.
Which is just incredible engineering.
It's wild.
Yeah.
And it weaponizes the tumor's own biology against it. But to understand the weapon, I feel like we first need to look at the environment, right? Specifically, we need to talk about fat.
Yeah, fat is the key here. So to understand this, we have to visualize what we call the tumor microenvironment, or the TME. Aggressive tumors, they build this complex ecosystem around themselves.
Like a little fortress.
Basically, yeah. And one of the defining features of that ecosystem is a massive concentration of lipids, meaning fatty acids. It's a fatty acid-rich milieu.
But why do they need so much fat? Because I usually think of cancer as being really hungry for sugar.
Well, they are, but think about what a cancer cell has to do constantly. It has to divide rapidly, right?
Good.
To turn one cell into two, it has to construct an entirely new outer cell membrane.
Oh, right.
Plus all the membranes for its internal organelles. And those membranes are constructed primarily from lipid bilayers. So the tumor demands just massive amounts of fatty acids.
So it's building material.
Exactly. It's so desperate for it that tumors will literally force adjacent fat cells, adipocytes, to break down and donate their stored fat directly to the tumor.
Wow. So the tumor is essentially strip mining the surrounding tissue for building materials.
Okay.
And then a surgeon's scalpel comes in. And when you physically cut a tumor out, you are inevitably rupturing a ton of cells.
You are. The mechanical disruption bursts millions of cells wide open, and this spills all their internal contents, including huge amounts of these free fatty acids, directly into the surgical cavity.
Okay, let's unpack this. It's basically like a microscopic oil spill, right?
And that is a great way to picture it, actually.
You would suddenly have this deep well of free-floating fatty acids just coating the wound. And I get that the residual cancer cells can use this to build new membranes, but the study notes this fat also acts as an active shield against the immune system. How exactly does floating fat stop an immune response?
Well, it comes down to how that fat interacts with macrophages.
Okay.
Macrophages are a type of white blood cell. Usually they're your body's frontline defenders. They patrol around, engulfing and digesting cellular debris, foreign substances, cancer cells.
The good guys.
Right, the good guys. And this active fighting state is biologically referred to as the M1 phenotype.
Okay, so we have these active M1 macrophages patrolling the surgical area, but they are suddenly swimming in this massive oil spill left by the surgery.
And because they are scavengers, they start absorbing those fats. The fatty acids accumulate inside the macrophages as tiny concentrated pools called lipid droplets, or LDs.
Oh.
And this physical accumulation of fat sets off a chemical chain reaction. The lipid droplets activate specific metabolic pathways, specifically the PTAR and AKT signaling pathways.
I want to pause on those pathways, PPAR and AKT, because it's not just that the macrophages are getting, sluggish from eating too much fat. This is an active reprogramming of the cell, isn't it?
It is a fundamental shift in their identity. Those signaling pathways physically and chemically reprogram the macrophage. It shifts it from the fighting M1 state to a corrupt immunosuppressive state known as M2.
So they flip sides.
They absolutely do. Instead of attacking the residual cancer cells, these M2 macrophages actually release signals that tell the rest of the immune system to just stand down.
Oh, man. The cancer spill fuel literally bribes the immune cells into protecting it, like corrupt security guards.
That's exactly what they are. And there is solid clinical proof backing this up in human patients, too. The researchers looked at the TCGA SKCM database, which is this massive genetic database for melanoma.
And what did they find in the human data?
The survival data is incredibly telling. They looked at genes responsible for clearing cholesterol and lipids out of cells, genes like ABCA1 and ABCG1.
Okay.
Patients whose tumors had low expression of these fat-clearing genes had significantly shorter survival times.
Because the fat was just hanging around doing damage.
Right. And conversely, they looked at genes that create proteins to stabilize and store fat droplets inside the cell, specifically a gene called PLN4. High expression of those fat-storing genes correlated with much poorer prognoses.
Let's do a quick recap here just to make sure we're tracking all these details. So we've got a surgical wound, it's filled with residual cancer cells, and it's flooded with fat from the surgery, this oil spill.
Yeah.
That fat gets eaten by our macrophage security guards, building up as lipid droplets. That activates the PPAR and AKT pathways, flipping them into corrupt M2 guards that protects the tumor.
You nailed it. And the human genetic data proves that when the local environment is bad at clearing fat or highly efficient at storing it, the cancer survives and the patient suffers.
Which naturally led the research team to this really fascinating hypothesis. If fat is both the shield and the fuel for residual cancer, what happens if you could turn that specific fuel source into a bomb?
Yeah, that's where the bioengineering really comes into play. The researchers built this two-pronged biochemical weapon. They call it a nanomodulator, and it's named R at LOX.
R at LOX.
Right. It uses two main ingredients to attack this fatty environment, rapamycin, which is the R, and lipoxygenase, which is the LOX. Let's start with lipoxygenase.
Okay, so LOX. What does LOX do in this toxic soup?
LOX is an enzyme. That means it acts as a catalyst to speed up specific chemical reactions. Its entire job in this scenario is to take those abundant extracellular free fatty acids, our oil spill, and catalyze them into highly reactive molecules called lipid peroxides, or LPOs.
And this isn't just about starving the cancer by taking away its fat, right? Converting those fatty acids into lipid peroxides triggers this very specific catastrophic event in the cancer cells called ferroptosis.
Yes, ferroptosis, which is fascinating because it is completely different from apoptosis.
Right, because apoptosis is that programmed, orderly cell death we usually hear about.
Exactly. Apoptosis is neat and tidy. Ferroptosis is messy. It's an iron-dependent form of cell death characterized by overwhelming oxidative damage to the cell's lipid membranes.
The analogy that really helps me visualize ferroptosis is just microscopic rusting. The LOX enzyme basically takes the tumor's own fatty fuel, oxidizes it, and turns it into toxic rust that literally shreds the cell membranes from the outside in.
It's a brutal mechanism, and it's highly effective because it's using the cancer's own excess resources, its own hoard of fat against it. But LOX is only half of the nanomodulator. We still have the problem of those corrupt M2 macrophages.
Right, the security guards that are full of fat droplets and protecting the tumor. The LOX is rusting the cancer cells, but we need the immune system back online. So that is where rapamycin steps in.
Exactly. Rapamycin is this well-known compound that inhibits a cellular signaling pathway called MTO. When you inhibit MTOR in a macrophage, you fundamentally alter its metabolism.
In what way?
You force that cell to start burning its stored lipid droplets for energy.
Oh, wow. So you're cutting off the chemical bribe by forcing the cell to just incinerate the fat it hoarded.
That's it. And as the macrophages burn through those lipid droplets, the M2 signaling pathways shut down. The cells physically revert back to the fighting M1 state.
They wake up.
They do. And the researchers proved this by tracking protein markers on the cell surfaces. After introducing rapamycin, they saw a massive spike in markers like CD86 and NOS2.
And those are specific to the good guys.
Yeah, those markers only appear on active, fighting M1 macrophages, so they knew the reprogramming worked.
Okay, so LOX shreds the cancer cells by turning their fat into rust, and rapamycin forces the immune cells to burn their fat so they start fighting again. That is brilliant.
But here's where it gets really interesting. There's a synergistic twist here. Rapamycin actually pulls double duty. It doesn't just work on the immune cells, it also targets a critical vulnerability inside the cancer cells themselves.
Wait, really? What's it doing inside the tumor?
This is arguably the most elegant part of the whole design. Cancer cells actually have a natural defense mechanism against ferroptosis. Because they constantly metabolize lipids, they're always at risk of accidentally oxidizing themselves.
Makes sense, yeah.
So they produce this antioxidant enzyme called GPX4. GPX4 neutralizes lipid peroxides. It basically acts as a chemical shield or a fire extinguisher against the rusting process.
A shield that rapamycin apparently knows how to break.
Yes. Inside the tumor cells, rapamycin triggers a process called autophagy. Autophagy literally translates to self-eating. It's how a cell recycles its internal components. Rapamycin tricks the tumor cell into eating and destroying its own GPX4 shields.
So let's do another quick recap of the battle plan here. We've got LOX outside the cell, aggressively generating all this toxic rust from the oil spill.
Right.
And at the exact same time, we've got rapamycin inside the cell, disarming the fire extinguishers, destroying the GPX4. And on top of that, rapamycin is waking up the immune guards.
Exactly. And the mathematical analysis the researchers ran showed that putting these two drugs together isn't just an additive effect, it is highly synergistic. They dramatically multiply each other's lethality against the cancer.
Okay, we have this incredibly lethal, perfectly coordinated biochemical weapon, but I keep coming back to a glaring mechanical problem here.
The delivery.
Yeah. A surgical wound is not a smooth, clean bowl. It is jagged, it's irregular, and it's full of microscopic crevices where these residual cancer cells are hiding out. If you just spray a liquid medicine in there, it's just going to wash away.
It would. And if you try to use traditional thermoresponsive hydrogels, you run into a totally different problem.
How so?
Well, standard hydrogels are designed to be liquid at room temperature and turn solid when they hit human body temperature.
Oh, I see. So the moment you inject that into a warm surgical cavity, it solidifies almost instantly.
Right. It just sits there as a rigid glob in the center of the wound, completely failing to seep into those deep microscopic pockets along the margins where the cancer is actually hiding.
So the physical delivery mechanism is just as important as the drug itself. If it doesn't reach the margins, it's useless.
Precisely. So to solve this, the researchers engineered what they call a light-driven thermotropic hydrogel, or LTG.
Let's look at the chemistry of this gel, because it is remarkably clever.
Mm-hmm.
It starts with a base matrix of gelatin and hyaluronic acid for structure, right?
Yes, that gives it its basic form. But the crucial innovation, the real magic, is the addition of tannic acid and iron ions, specifically F3 plus ions.
Yeah, 3 plus 4.
Right. When you combine tannic acid and these iron ions inside that gelatin base, they form a metal polyphenol network within the gel.
So the best way to picture this metal polyphenol network is to imagine millions of microscopic solar panels embedded evenly throughout a thick wax.
That's a perfect analogy.
So if you just inject this firm gel into the wound at room temp, it doesn't flow. It's solid. But then the surgeon takes a near-infrared laser, which, by the way, is completely harmless to human tissue.
Right, it passes right through.
They shine it through the skin directly onto the surgical site. What happens then?
The near-infrared light hits that metal polyphenol network. Those little solar panels capture the light energy and rapidly convert it into heat. The localized temperature of the gel rises to about 43 to 50 degrees Celsius.
And that specific temperature is the trigger.
Exactly. It triggers a gel-to-sol phase transition. The solid hydrogel actually melts into a highly fluid liquid state, a sol. And because it's now a liquid, it flows perfectly.
It just adapts to the exact irregular geography of the wound.
Yes. It seeps deep into every single microcavity along those invasive margins. It literally casts a perfect mold of the wound inside the body.
That is so cool. And then the surgeon simply turns off the laser, the gel cools back down to normal body temperature, and it locks back into a solid state.
And once it solidifies, it becomes a sustained drug reservoir.
So let's recap this part. We inject a firm gel. We melt it with a laser so it flows into every microscopic crack.
Yeah.
We turn the laser off, and it hardens perfectly in place.
Right. And because it's now perfectly molded to the tissue, it slowly and continuously releases the R at LOX nanomodulators right at the boundary where the residual cancer cells live. It maintains this targeted treatment for over 48 hours.
Plus, the laser heating process isn't just a physical switch for the gel, is it?
No, it has a therapeutic effect too. Raising the temperature of that microenvironment to around 50 degrees Celsius induces mild hyperthermia. That heat physically damages and bursts open residual cancer cells right on the surface.
Which spills even more fatty acids into the environment for the LOX to turn into toxic rust.
Exactly. It becomes a fully multimodal therapy.
It's just an incredible sequence of events.
Yeah.
But, Translating a brilliant bioengineering concept from a petri dish to a living organism, that is always the ultimate test.
Oh, absolutely. The in vivo trials are where a lot of great ideas fail.
So when they moved to the animal models, how did this actually perform?
Well, they tested this on post-surgical models for both melanoma using C57BL6 mice and breast cancer using the 4T1 Luke model.
And both of those are notorious for being aggressive, right?
Very. They specifically chose highly aggressive cancer strains known for thriving in lipid-rich environments and having just notoriously high recurrence rates after surgery.
The baseline survival data for the control groups really highlights how aggressive they are. I mean, for the melanoma model, the mice that underwent surgery but received no hydrogel treatment, they suffered rapid tumor recurrence.
Yeah, every single one of those control mice died within 10 days.
10 days. But when they applied the full treatment, the LTG hydrogel, the near-infrared laser activation, and the R at LOX delivery, the median survival time nearly doubled.
It did, and the physical recurrence of the tumors was severely suppressed.
And the breast cancer model was even more striking, wasn't it?
It really was. Over 90% of the treated subjects in the breast cancer group survived beyond 45 days, with almost zero signs of the tumor returning.
That is a massive difference.
It's night and day. And to verify the actual mechanism behind the survival, the researchers extracted the surrounding tissues and used flow cytometry. They wanted to sort and count the individual immune cells to see if the TME, that tumor microenvironment, had actually been remodeled.
So they were looking for our security guards, the macrophages. Did the rapamycin actually wake them up in a living subject?
It definitely did. The flow cytometry data showed a massive drop in the corrupt immunosuppressive M2 macrophages. And that was paired with a huge surge in the active, fighting CD86 positive M1 macrophages.
So the guards woke up.
Yeah.
And once those frontline defenders were back online, they started calling for backup.
Exactly. They saw a massive infiltration of CD8 positive T cells into the surgical cavity.
These CD positive T cells, those are the immune system specialized assassins, right?
Yeah. Their primary function is to hunt down and eliminate cancer cells. So the treatment didn't just stall the immunosuppressive environment, it entirely reversed it into an active, highly hostile environment for the cancer.
Now, I have to push back on one aspect of this because it sounds almost too perfect.
Sure. What's the catch?
We are talking about injecting a chemical gel into a fresh surgical wound. We are blasting it with a laser to heat it up to 50 degrees Celsius. We are deliberately causing catastrophic oxidative rusting through ferroptosis, generating all these toxic lipid peroxides. How is the healthy skin and tissue surrounding the wound not completely destroyed? Aren't we just trading a tumor recurrence for a severe chemical burn?
It's a really valid question. Toxicity is usually the limiting factor for any cancer therapy. You want to kill the cancer without destroying the host.
Right.
But the pathological analysis of the surrounding healthy tissue, they used H&E and Masson's trichrome staining. It revealed something completely counterintuitive. The treatment didn't just spare the healthy tissue, it actually accelerated wound closure compared to the control group.
Wait, really? How does creating toxic rust accelerate healing?
Well, it has to do with the highly targeted nature of the mechanism. Remember, the ferroptosis is driven by the LOX enzyme acting on the massive concentration of fatty acids spilled by the cancer cells.
The oil spill.
Right. Normal, healthy cells aren't bathing in that extreme lipid environment, and they aren't rapidly dividing like cancer cells, so they aren't hoarding fat.
So they don't have the fuel to burn.
Exactly. They are relatively spared from the rusting effect because they don't have the lipid profile to trigger it. The weapon basically only arms itself in the presence of the cancer's unique environment.
That's incredibly elegant.
It is. And furthermore, the mild hyperthermia from the laser, combined with the sudden clearance of that immunosuppressive fog by waking up the immune system, it actually promoted healthy tissue regeneration.
So the body could finally heal itself.
Yeah, the histological staining showed enhanced angiogenesis.
Which is the formation of new blood vessels.
Right. And it also showed faster reepithelialization, which is the growth of new skin over the wound. It eliminated the residual cancer while actively healing the surgical trauma.
It is a brilliant example of taking the unique, chaotic metabolic signature of a disease and using it as the activation key for the cure.
That's a great way to summarize it.
Well, we have covered a massive amount of molecular biology and materials engineering today, so let's just do one final recap of the whole journey. We started with a fundamental problem. A surgical cavity flooded with tumor-feeding fatty acids that bribe our immune system into submission.
The oil spill and the corrupt guards?
Right. To counter it, researchers engineered a hydrogel packed with a metal polyphenol network that absorbs laser light, melts into a fluid to fill every microscopic crevice, and then locks into a solid reservoir.
The perfect mold.
And that reservoir then deploys a deeply synergistic attack. It releases lipoxygenase, LOX, to turn the excess fat into toxic rust, shredding the cancer cells through ferroptosis.
While simultaneously releasing rapamycin to destroy the tumor's internal antioxidant shields, the GPX4, and forcing those corrupted macrophages to burn their fat and reawaken the immune system.
It takes the messy, murky reality of a post-surgical wound and turns it into a highly controlled, localized trap.
It really does. And, this raises a fascinating question about the future of targeted medicine in general.
Oh.
Well, we've just seen how a localized, heat-triggered hydrogel can weaponize a lipid-rich environment to destroy cancer. But cancer isn't the only disease defined by dangerous accumulations of lipids.
That's true.
Consider atherosclerosis, right? Where fatty plaques build up and harden inside our arteries.
Oh, wow.
If we can engineer these smart hydrogels to seek out, bind to, and melt away localized fat deposits using harmless external lasers, could a similar phase-shifting technology be injected into the bloodstream?
To selectively clear arterial plaques without needing invasive cardiovascular surgery.
Exactly. If we can hack the fat in a tumor, what's stopping us from hacking the fat in a blocked artery?
That is a massive paradigm shift. Instead of viewing a chaotic, disease-heavy microenvironment purely as a barrier, we can actually engineer therapies that require that specific chaos to function.
It completely flips the script on how we treat complex diseases.
It really changes how you look at the complexities of the human body. Well, thank you for joining us on this deep dive. Keep asking questions, keep looking past the obvious, and we will see you next time as we continue to explore the incredible mechanisms driving modern medical breakthroughs.